great opportunity to present our recent innovation in the field of spatially resolved proteomics. This is a very new technology that's been developed in a close collaboration with Buchanan Lab here at Westlake University. And today I'm very excited to present this technology for the first time to the broad audience. And as mentioned before, the title of my talk will be Spatially Resolved Proteomics via Tissue Expansion. But before we dive in actual technology, I would like to spend a little bit more time on taking a broader look at the technologies that we used before for molecular profiling of a tissue. And of course, we know in the past 2 decades, genomics and transcriptomics launched the precision revolution. And particularly in the past 2 decades, these technologies have yielded some breakthrough markers for risk prognosis and prediction of drug response and treatment. And however, as we are getting more and more data, we also start to realize that not all genomic and transcriptomic markers demonstrate outstanding ability to predict treatment outcomes in the clinical studies. Individual genetics variations, such as single nucleotide polymorphism, generally accounts for a very modest proportion of phenotypic variability. It's usually less than 10%, and relatively small increments in risk, less than 1.5-fold. In most of the cases, this is not enough to justify clinical intervention or changes in treatment. While genomics defines potential gene products, proteomics better reflect the actual phenotype. And recent proteomic characterization of more than 600 cancer cell lines with previously published genomic transcriptomics and drug screening data showed that, for example, protein expression data correlates significantly better with drug sensitivity or resistance than genomic data or even transcriptomics data. Therefore, in the recent years, the proteins are gaining more and more attention as both pathological biomarkers and drug targets. And one of the major goals of a modern medicine is to reveal the correlation between pathological conditions and proteome perturbations. However, in most of the cases, we usually don't know what specific protein or protein biomarkers are involved in the various pathologies. And one challenge of proteomics we have to deal with is extreme proteome complexity. And this complexity arises from the fact that each gene produces several protein variants, far beyond what we can predict from DNA or RNA sequences. And while there are only about 25,000 genes in our genome, it results in more than 1 million of proteoforms. And also these proteoforms interact with each other in a very complex way. So a single cell can easily encompass hundreds of thousands of different proteoforms, reflecting the chemical diversity of protein molecules, each having the potential to alter cellular structure, function, and interaction. Another challenge of proteomics is that, unlike nucleic acid, RNA, and DNA that can be amplified in order for their detection, proteomes in the biological samples cannot be amplified. It means that we also have to deal with a far less material for identification. So proteome complexity also justifies untargeted proteomics as one of the most efficient and well-established methods for biomarker discovery. And over 80% of studies for new biomarkers discovery actually were based on untargeted proteomics technologies, while a very small proportion of them, less than 9%, use exclusively targeted proteomics. And mass spectrometry is the most powerful technology for untargeted proteomics. And MS-based proteomics provide an advantage over antibody-based technologies and fluorescent proteins in that it's highly multiplex and requires no prior knowledge of protein targets. It's also retained information about truncated or post-translationally modified proteoforms. In particular, data-independent acquisition using liquid chromatography tandem mass spectroscopy offers a superior depth and throughput of proteomic analysis among all mass spectrometry-based methods. And most of the proteomic studies are done on tissue as heterogeneous mosaic of cells in the different pathological stages. So this creates a pressing need to perform proteomic analysis on with a specially resolved mode, when we can distinguish nearby cells one from another. And if we look carefully, we'll see that bottom-up proteomics is the most sensitive method for untargeted proteomics. And there are several ways to perform specially resolved proteomics using bottom-up methods of mass spectroscopy. One, although not very popular way to perform specially resolved proteomics with a resolution about 250 microns is a deposition of a hydrogel that been soaked in a trypsin directly on the tissue samples. After the trypsin digests and extract the peptides from a tissue, this hydrogel disc are collected and extracted for mass spec analysis. There are several problems. Although it's a very simple method, it has several limitations. First of all, the resolution is only 250 micrometers, and also the duration of extraction is more than 24 hours. Another thing, that we cannot perform the very tight coverage of entire tissue because this hydrogel disc has to be specially separated. An alternative method to perform specially resolved proteomics is liquid extraction surface analysis. While this method is significantly faster than hydrogel extraction, it takes about 10 minutes per spot, it has two other limitations. So first of all, the spatial resolution is very low, it's about 500 microns. And at the top, it requires complicated and custom-built setup in order to perform the liquid deposition and extraction from a tissue surface. And the most popular methods for specially resolved proteomics that provides the highest resolution down to a single cell is laser capture microdissection, when the tissue is dissected using a high power beam of a UV laser with a precision down to a single cells under microscope system. The time that required to dissect the tissue can be as little as several hours. And this method is actually the most popular method among all this specially resolved proteomics method. If we see the number of publications that using LCM, it will be over 90% of all publications using LCM for specially resolved proteomics. However, there is also several other limitations of laser capture microdissection. So first of all, it's significantly, it requires the complex and expensive equipment, such as the microscope with a laser, and the throughput of this method is also quite limited. Here's an example of microdissection being performed in real time, where the operator has to manually draw the area to be extracted. Then the laser goes and cut it out from the tissue, and then operator needs to confirm with this piece of tissue being physically removed and transfer it to the cap of a tissue of a tube for further peptide extraction. So this is a recording being done in real time, and you can also see that it's quite complex and laborious procedure. There is another limitation here, that after microdissection, specifically single cell samples, the peptide extraction has to be done in nanovolume in order to minimize the loss of the peptides. And another technology that coupled with this is nanopods technology, that also require quite sophisticated and complex equipment, which is not commercially available. So LCM, although is the most widely used way to perform spatially resolved proteomics, it still has multiple drawbacks and limitations. So first of all, it requires expensive and sophisticated equipment. LCM has a very low throughput and requires very laborious procedure where the operator has to manually confirm each piece being extracted from the tissue. Trace samples that being excised by LCM has to be processed through a laborious and time-consuming process. And also efficiency of peptide extraction requires custom-built equipment for nanoliter sample handling and processing. So we decided to overcome this limitation of LCM by thinking from a completely different perspective. We decided, what if we can do microdissection of the samples after its physical magnification, right? If there is something very small, can we make it bigger so it's easier to dissect, for example, using very simple scalpel or blade in your hands. And one of the way to increase in size the tissue is the technology that I've been working during my postdoc at MIT that we called expansion microscopy. An idea of expansion microscopy was physically magnify the brain, the biological tissue that's been embedded in hydrogel, so we can image it with super resolution under conventional microscopes. And here you saw the real-time expansion of a single tissue brain slice, but after expansion we can put under microscope and see and visualize the nanostructures under conventional microscope because they're bigger than the diffraction limit of a microscope. Here is an example of a confocal image of a brain ball labeled brain tissue, and what you see actually here is that the colors are coming from fluorescent proteins. What it means that proteins are retained in expanded tissue, and they not only retain, but they also preserve their functionality. In this case, they are fluorescent. So before moving on, I would like to introduce a little bit more of details of this technology and how the physical magnification of a sample is happening. When we start working on this project more than 6 years ago, we came across smart polymers. With smart polymers can expand in a pure water up to 1,000 times in volume. This polymer is called super absorbing polymers. It's the same polymer that you can find, for example, in baby diapers or in the water expandable toys. When you put it in the water and they expand up to 10 times in linear dimension or up to 1,000 times in volume. So the expansion of this molecules happening due to the water molecules is dragged inside of a polymer chains through a process called osmosis, because of a polymer consists of a very simple chemicals that contains the hydroxylic group and but being conjugated with a sodium atom. When we add water, the sodium ions are shielded and negatively charged carboxylic groups are repelled from each other, making the polymer chains to repel, to spread out. And if we zoom into molecular level, we will see that in a collapsed state, the polymer looks like a very dense network of a polymer chains. You see them as a wide structures. And when we add water, water gets in between these polymer chains, push them apart, making them to expand. And we decided, what if we can install these polymer chains into biological sample in a way where these chains can be attached to the biomolecules, and when we add water, the sample expand, the polymer network expand the sample taken biomolecule fluoride. And surprisingly, it can be done in 3 very simple steps. And here is step number 1. We modify the proteins in the tissue with a special handlebars. These handlebars will eventually be attached to the polymer chains and let the polymer chains pull apart the biomolecules. After the proteins inside of in situ been modified, we infuse the sample with a monomer, with the building blocks of a polymer chains, with the monomers. And these monomers, after infusion into the biological samples, can be very easily polymerized through radical reaction. We initiate the polymerization, and the growing chains of polymers can also covalently attach themselves to the biomolecules from the handlebars that we installed earlier. And now when we synthesize gel sample composite, we need just one more step. We need to add the pure water in order to let the polymer chains spread apart. However, here we also came up with one trick. Before we add water, we need to loosen up intramolecular connection between the proteins, and for this we perform a very simple step that we call homogenization, mechanical homogenization of a sample, so the sample does not resist to the expansion. So what we do, we break out the interaction between the proteins, and after interaction between the proteins being loosened up, we add the pure water. And you see that now when the polymer chains are expanding, we're taking the molecules, the proteins, for a ride. And every molecule here is covalently attached to a polymer network. And what's great is that after expansion, the relative location of all biomolecules remain the same. Here's an example of a brain slice from mouse that had been expanded in a hydrogel, as I just described, and stained with a very simple colorimetric dye, Kuma-C blue. It's the same dye that we use to stain protein gels. So the gel that we develop can expand the biological sample up to 7-fold in linear dimension. So here, with two slides, it's the same brain slides before and after expansion being shown in scale. And as you can see, the staining with Kuma-C preserves and allows us to visualize morphological features of a tissue. So, for example, experienced neuroscientists can easily identify different morphological regions here. And if you're not very experienced, you can easily use the brain atlas for visualizing the morphological boundaries of this tissue. So once tissue is expanded, we know that proteins remain inside. We can easily, now when the sample is 7 times bigger, now we can easily take, for example, a punch, remove a piece of tissue from a slice at the location we want to manually, get this piece of gel with our sample, and extract the peptides. This is extremely simple and straightforward procedure, and here's an example of microdissection, the voxelation of a tissue using a microdicer. So once the tissue is expanded, we dissect it in a small voxels. So this is a real-time video that I just recorded before the talk. When we take a we take a tweezers, but now we can remove the piece of tissue from a given brain region. And the dimensions of a piece of gel I'm removing right now is 2 by 2 millimeters, and 2 by 2 millimeters before expansion been roughly 300 microns. So I just did microdissection 300 by 300 microns in less than a minute with my hands, with a high precision. And also you can see there's no special equipment required. So, of course, once we remove the tissue, the first thing that we've done, we perform very simple in-gel digestion procedure, very similar procedure to what done usually to page gels, protein gels to extract the proteins. Here's an example of a brain tissue punched with a blade. We extracted the proteins and put it into mass spectrometer, and what we immediately saw, that we can efficiently extract the peptides, and these peptides can be easily identified into a proteins. So We also observe pretty small miscleavage rate in these gels, less than 14%, while preserving very high yield. These encouraging results allow us to develop a very simple, straightforward protocol for spatially resolved proteomics. And here is a workflow. So we take a brain tissue. This brain tissue can be actually stained with antibodies or with other dyes. We add a protein anchoring to it. This is procedure that I showed you earlier. After the protein has been anchored, we embed the sample into a hydrogel. After homogenization, the hydrogel is expanded, stained with a Kumasi blue stain, allows us to visualize it by naked eye. We image it in order to have a map of expanded sample. We perform manual microdissection with a precision down to 100 microns. Extracted or excised pieces of gel being moved to the PCR tubes for proteolytic sample preparation, extraction of the peptides, and running it on LC-MS machine. So this is the chemical transformation that happening to the protein. So first, the protein anchoring happening through the primary amine group, where we add a group that can interact, that can actually participate in the polymerization reaction. However, this amide group that formed, that links the proteins to the polymer chains, can be easily hydrolyzed during in-gel digestion. So the anchoring of a peptide is reversible. So I want to also mention very quickly a few, just give a little bit more details on how fast is this process. This process takes about, in total, about 55 to 58 hours, starting from a fixed sample. However, most of the time is incubation with different solutions, while hands-on time for this process is only 5 hours. And what's important, many samples can be processed in parallel, unlike for other methods that used for spatially resolved proteomics. Of course, we need to perform the benchmarking of a new technology, and for benchmarking, we choose to compare it to two golden standards methods for peptide extraction from a tissue that have been used in proteomics. It's in-solution digestion is the most trivial way, and pressure cycling technology that I abbreviate here as PCT. So, and here you can see that the protein yield actually achieved with the proteomics being almost 50% higher than the golden standards in the field. And another interesting thing that we've also observed, reduced miscleavage rate during trypsinization. So it was miscleavage rate for proteomics was about 20%, while for PCT and in solution was on a level of 25 to 27%. We believe that this is actually happening due to the fact that then the protein are anchored into a hydrogel, they expose to the trypsin from all sides, so trypsinization can happen with higher efficiency. Another important parameter is protein and peptide identification. So we compared number of peptides and protein that can be identified from a comparable size of the samples. Although we do see a smaller number of peptides and proteins being identified from the same sample, in case of proteomics and PCT methods, the decrease is less than 10%. Yes, it's statistically significant, but it's not biologically significant. It's also important to show that blank gel and MS buffer show basically noise in mass spec analysis, demonstrating that there is no diffusion of a protein throughout the gel. The proteins still remain tightly attached to the gel during the microdissection. So there is no artifacts. And both, all methods show very comparable overlap of identified proteins. So 67% of all proteins identified in overlap from all three methods. And another important thing that the methods were not, the proteomics was not biased on the subcellular localization of proteins that had been extracted. So efficiency of extraction from all cell compartments is comparable to golden standards in the field. So although we did see a lower number of peptides and proteins identified in case of proteomics, this does not affect application, for example, for biomarker discovery, because we saw less than 50 biomarkers being identified in proteomics. So while in solution NPC identified about 500 biomarkers, it was identifying more than 450 biomarkers in case of proteomics. As I also mentioned, protein localization is comparable for all three methods, and protein types also comparable for all three methods. So whatever data I presented till now was done on a brain tissue, but also we need to make sure that this technology is generally applicable. And another thing, that since we are mapping back the identified proteome to the tissue morphology, we have to confirm that tissue expansion happens isotropically. Here's an example of a tissue expansion that we performed using proteomics workflow for 3 different types of tissue. It's a mouse brain tissue, mouse liver tissue, and mouse breast cancer. In all three, for all 3 tissues, we quantified the distortion that's been introduced by expansion by calculating the length error of an expanded sample that been non-rigidly registered on pre-expansion sample. And what we observe that on a scale of about 1.5 millimeters, we see the distortion rate less than 80 microns. So the distortion is significantly lower than a special resolution that we use for microdissection. So we can say that in this case, proteomics does not introduce any distortion into the tissue morphology. Another important parameter we wanted to test, how small can be the spatial resolution of a proteomics while giving us reasonable number of identified peptides and proteins. So here we perform a special microdissection of a brain tissue at different resolution in the range from 120 microns to 730 microns. And what we saw that on a level of 120 microns, we still were able to identify around 1,000 proteins. And it's important also to note that the volume that we used for sample preparation was less than 1 nanoliter, which is more than 200 times smaller than the minimum volume that's used for PCT and more than 1,000 times bigger than usually volume used for in-solution digestion. So we're showing that the proteomics can operate on a 100 micron spatial resolution while providing significant number of identified peptides. Now it's important to put this technology into the context with other methods for spatial resolution that I presented earlier. And you can see here, for example, although we're still not reaching the spatial resolution of LCM, nanopod method is actually, which is right now is the characterized by highest performance in terms of spatial resolution and identified proteins. We are outperforming other methods such as LESA and LCM plus in-solution digestion by more than 5-fold. So we believe that this technology actually provide new modality into a specially resolved proteomics. So, and one of a demo that we would like that we perform using this technology in order to identify some biological biomarkers for Alzheimer's disease, we compared the proteome profiles of brain tissue in wild type and Alzheimer's disease mouse model. In this case, we use APP-PS1 mouse, and we compare 2 groups, old and young animals, 5 months and 18 months. So early stage and late stage of AD development. So in this case, we were able to manually perform microdissection of 5 different brain regions, dissecting visual cortex, hippocampal brain region CA1 and CA3, and data gyrus with a high precision. So we were able, after punching the brain tissue, we were able to image it and confirm that all the brain regions that we've been excised actually located in the right place. So after analysis, we found out that the, not surprisingly, young and wild type NAD mice did not differ much in their proteome. It's only been targeted by 2 proteins that actually been mutated. However, we saw a significant difference in the proteome of old wild type NAD mice. Specifically, the area that being the most affected by AD was hippocampal CA1 area, where we identify more than 198 differentially expressed proteins. This results actually confirmed from the literature that hippocampal area CA1 is the most affected brain region in Alzheimer's disease. We were able to further pinpoint the proteins that have been actually upregulated and downregulated in AD mouse. So while we confirm, for example, from previous literature results that ApoE protein and syntaxin protein being the most affected, we also find out 2 new proteins that have not been reported before involved in the AD, such as proline-rich protein 7. So, and while I've been mostly talking about proteins here, I also would like to briefly mention another interesting part of this technology, but this technology can be also versatile. The chemistry that we use can be easily extended beyond the proteins. And here's an example of brain tissue that's been expanded using the hydrogel while preserving both proteins and mRNAs inside of expanded tissue. So by swapping or by adding an extra chemical anchors, we also can modify the nucleic acids that can be preserved in a tissue. So in this case, we can perform multi-omics, truly multi-omics analysis, when we can identify both mRNAs and proteins in the same sample. And this is some future direction of this technology. And of course, this is, of course, I would like to acknowledge the members of my lab, molecular bioengineering group. It's Ciji, Xiaoqin, and Hanbin, who help develop this technology, and of course members of Guomics lab who are co-inventors of these technologies. And first of all, Tianan, Judy, Lilu, Yaotin, Zhendong, and Yan Zhou. And this work being supported by Natural Science Foundation of China and Brain and Behavioral Research Foundation, as well as Westlake Educational Foundation. So thank you very much for your attention, and I will be happy to take any questions regarding our technology.
Okay, thank you so much for giving us such a wonderful talk. So now let's see what kinds of questions we've got here in our chat box.
All right.
Shuchang, will you please just...
Yeah, so the first question is about after you expand the samples, could the samples be shrink or contract to the origin?
Yes, so yeah, absolutely. The hydrogel can be expanded can shrink back to its original size by simply changing the osmolarity of a buffer. So if we add a little bit of a salt, such as sodium chloride, it will shrink back into original size. Yes. And another thing, even we not only can shrink it back, but we also can store it for a significant amount of time without any degradation in a shrink stage, yeah, in a shrunk form.
Mm-hmm. Okay, thanks. So the second question is about what's the success rate of user expansion? Like, will you fail to expand your samples?
So we mostly, as I showed the three examples of a tissue, liver, mouse, and cancer, and I would say the success rate is 100%, unless you forget to add the chemicals. Yes, so one important part of this technology is very simple technology. So many people who come into my lab can pick up gel expansion part. Expansion itself is just a series of adding and removing the chemicals, adding chemicals to the brain or to the tissue and removing chemicals from a tissue. So it's pretty straightforward procedure that does not require any prior training, as long as you can add chemicals and remove chemicals from a sample.
So does that mean if you just follow the protocol, you will succeed?
Oh yeah, you just started. You just started doing this procedure, like
yeah. It's very
what was it your success? How's your success rate?
I think it's at first it may fail some samples, but it's very straightforward.
Exactly.
It's
yes.
very
So as I show on this slide here, the timeline is of a procedure is about 55, 58 hours, including mass spec acquisition. So in principle, the expansion can be done in 2 days. All the chemicals that we're using in this procedure are available, commercially available, just off the shelf, and they are pretty cheap. I would say this technology is cheaper than any other specially resolved proteomic approach with out there. It's very, very affordable. Yes.
Yeah.
It does not require any specialized equipment on a sample preparation step. Of course, you need a mass spec machine, but on a sample preparation step, it does not require any special equipment, training or knowledge or background. Anyone can do it.
So the last question is about what's the limitation of detection of proteomics?
Limitations.
Yeah.
So the smallest size at the moment that we tried was 120 micron, and the major limitation here that we couldn't handle the smaller pieces of a gel just because they are hard to see by eye. And second, the second limitation is efficiency of extraction of a peptide. So, and actually, this is a limitation for any other specially resolved proteomics. You know, like if you take 200 cells, right, and together in a bulk, extract the protein and take amount of a protein that corresponds to one cell out of it, you will get a pretty good peptide identification. But if you just take one cell and extract the peptides, you're going to get very less because of the losses in the sample preparation step. So this is basically the same limitation as for other methods. It's efficiency of sample preparation to make sure that the peptides are not get lost on the way during multiple washing steps and sticking to the surface of the tubes. Yeah, you handle it. So, but in principle, at the moment, we are on 120 micron, but we believe we can go lower. Lateral resolution, yes. Any other questions?
No. Thanks.
All right. Okay. Thank you very much. Thank you.